Hello, welcome back. We are now in part 7 of 9, looking at biomolecules. You guys are probably sick and tired of biomolecules and biochemistry and all that kind of good stuff by now. So I want to start off kind of reviewing where we've been with our look at biomolecules and their organization. And then we're going to jump into the third class, which is proteins. And I'm going to be honest, proteins is a big section. In fact, I'm dividing this up over 2 videos because it is so big. Lots and lots of details within proteins. So let's start by doing a review. All right, so we've got, again, our first biomolecule that we looked at were carbohydrates. And we know that the monomer for the carbohydrate was a monosaccharide. And that this was forming a glycosidic bond. And when we talk about a glycosidic bond, we're referring to the use of 2 hydroxyl groups to go ahead and conduct dehydration synthesis. Again, remember, all of these are utilizing dehydration synthesis. All right. And then we have nucleic acids. Right, and we said that their monomer for nucleic acids was a nucleotide. Right, the bond that is created vertically, that's involving the functional groups, that was what we referred to as a phosphodiester linkage. And we said that that was formed between a phosphate group and a 5' carbon of one sugar and a 3' carbon of the sugar above it. All right, so that's where we've been so far. Where we are going now are proteins. All right. And what I will tell you is, as of right now, that a monomer of a protein is an amino acid. And the bond that we are going to be looking at creating is a peptide bond. Right. And we're going to leave this a mystery for right now. We will get into it as we go into the lecture. All right, but that's kind of a review where we have been and where we are going. Both of those things are critically important. We need to know where we've been and we need to know where we're going so we understand where we are now. And so with that, let's jump into it. As I've already mentioned, the monomer of a protein is an amino acid. All right. Lots of talk about amino acids. You hear them all the time. Dietary supplements, you hear it as far as concerns with vegan and vegetarian versus omnivore or more meat-based lifestyles, and how do you balance these things out. We hear amino acids from the perspective of health. We hear, you name it. We constantly hear about amino acids, and it's protein based. I mean, these are the building blocks. These are the monomers that make proteins. And proteins run and drive and direct every function within the body. We're going to talk about that. We're going to look at just how widespread proteins are throughout the body. Right? For our proteins, when we talk about what makes up our proteins, there are 21 different amino acids. That's what AA stands for. All right, not Alcoholic Anonymous or anything like that. Although by the end of this course, you might be needing that. This is amino acids. Right? This is referring to amino acids. And so 20 different amino acids, 21 if you consider one of the variables in there. exist that make up our proteins. Now understand, there are hundreds upon hundreds of amino acids. Right? But when we talk about making up our proteins, it's a combination of these 21 amino acids that really drive the building and the formation and the production of our proteins. And like other biomolecules, amino acids have a very common structure. Right? There is uniformity in how they look. And so part of that uniformity is that there is a central atom. Right? And that central atom is a carbon atom. I don't know about you, but I get tired of looking at red for a laser pointer. Let's do blue. Right? And so here is the central carbon atom. Now, coming off of this central carbon atom, we have two functional groups. We have a carboxyl group right here. Hopefully you recognize that. So here's a carboxyl group. Here is an amino group. Hopefully you recognized that. All right. These two groups are always there. In fact, when we talk about forming a peptide bond, it is the carboxyl group and the amino group that are engaged in forming the peptide bonds that are going to join amino acids. Right? So you need to be able to identify and recognize this. All right? Here's a hydrogen atom. He's always there. And then the variable in the mix is this R group, this side chain, right, or this reactatory group. This reactatory group, this R group, really defines how the amino acid engages or interacts with the environment around it. The amino group and the carboxyl group determine the peptide bond. The R group determines how do I, as an amino acid, function within the environment. Right? How well do I play with others within the environment? That's the job of the R group, to make that determination. What do I mean? Well, here's a summary table of all 21 amino acids. If you look at the basic structure of all of these amino acids, right, look at the base. This is all the base. It's identical. Right? It's a central carbon with a hydrogen, with an amino group, with a carboxyl group. That is consistent throughout all of these molecules. What varies on each of these amino acids is the R group. The R group is different on each amino acid. So if you were required to be able to know and identify each of these 21 amino acids, you would have to remember the functional groups, because that's what determines the amino acid. Now, you don't need to know that. You do need to know that the R group does determine reactivity, and how it interacts with its environment. In other words, the solubility. Is this going to be an amino acid that is readily going to be able to dissolve in water or react with water, a term that we refer to as being hydrophilic? Or is this going to be a molecule that is going to not mix with water, right, and therefore cannot be dissolved, and needs to be absorbed maybe through your diet in concentrations or in conjunction with high concentrations of fat, right, because it is lipid soluble. Right? In other words, hydrophobic. Hydrophobic is water-fearing. It usually mixes with oil. Hydrophilic is water-loving. It usually mixes with water and avoids oil. All right. So if we look at the first 9 amino acids, right, glycine, alanine, valine, leucine, isoleucine, methionine, which is a critically important amino acid, phenylalanine, tryptophan, which is the same chemical that does not put you to sleep when you eat turkey at Thanksgiving dinner. We'll talk about that later in the semester. And proline. Those 9 amino acids are non-polar amino acids. Right? Non-polar amino acids, which means they are not soluble in Water. In order to absorb these amino acids, you need to have fat in your diet, or you will not absorb those amino acids. So diets that remove all of that fat and expect you to carb load, you're actually depleting your body of these amino acids, which are critically important in the production of proteins. Now, the other thing that you need to remember about these 9 hydrophobic non-polar amino acids is that these guys are referred to as, they have a name. These guys are the essential 9. They are the essential 9 because they are only acquired through diets. You have to eat these. You have to consume these. You have to absorb these. Your liver cannot make them on its own. All of the other amino acids your body can actually make and produce on its own. But not these guys. They have to be acquired. And when you look at something like methionine, right, when you look at this guy right here, whenever you are doing protein synthesis, that is the first amino acid in producing the protein. And if you're lacking that in your diet, then you can't successfully make proteins. Which is where the balance comes in when we talk about people that are vegan and vegetarian. And the question is always, how do you get your protein? This is why. Because it's very hard in a plant-based diet to get adequate levels of protein. And I'm sorry, but taking a supplement, if you have to take a supplement to replace what you're not getting in your diet, the diet is probably not good for you. Because it's not all encompassing. Right? The fact that you have to take a supplement means that your diet is not complete. And so you got to balance that out. Okay, you got to balance that out. I'm not saying it's you shouldn't do it. I'm saying you need to be really aware of what you're doing because you could be doing more damage and harm than what you actually think that you're doing. All right. So kind of keep that in mind. Right? The next 6 amino acids, right? Serine, threonine, cysteine, tyrosine, asparagine, and glutamine, right? They are hydrophilic. They are soluble in water, and so those R groups allow it to react with water. Right? The bottom 5 are also soluble in water, which makes sense. They're more acidic and alkaline, and we know that acids give up hydrogen protons to the water, and we know bases accept protons, either from an acid or from the water. We know already by them being defined as an acid or a base or acidic or basic that they're going to react. We just talked about that when we looked at acids and bases and pH. And so things like aspartate and glutamate and lysine and arginine and histidine. By the way, histidine is a vitally important hormone, amino acid in the production of DNA. It's the histidine proteins, amino acids that actually allows DNA to twine, to wrap. They're called histones, but they're made out of histidine. So if you're lacking that, you're putting your body at a disadvantage to go ahead and duplicate and repair DNA. See how things can literally start unraveling here when you're not really understanding the biochemistry. Right? We jump on things because, everyone else is doing it. Or, this is horrible, I want to do this. And, okay, weigh it out. Weigh it out. Do better. # do better. Educate yourself. Sorry. I sidetracked. So, What's important for you to understand here? You need to understand that the R group determines reactivity. Right? You need to understand that the essential 9 are hydrophobic. They do not absorb in water. They do not dissolve in water, and that you need to acquire them in your diet, and that diet has to have a level of fat in it, or you will not absorb it. All right. Everything else there is hydrophilic. All right. So things that have a polar side chain will go ahead and react with water. Things that are acidic or basic because of the R group will react with water. This here is just a very simple slide, just kind of does some definitions that you probably already know. You already know what a monomer is. You already know what a macromolecule is. Right? We already know that dehydration synthesis is critical in this production. Right? And when we talk about polymerization, we're talking about that process of doing dehydration synthesis, of linking a monomer to a monomer to a monomer to a monomer. It's just another name for it. Again, there shouldn't be anything all too earth-shattering new on this slide, because we've been looking at the same thing over and over and over again. Right? We know that the monomers, the amino acids, are going to undergo dehydration synthesis. And when we do that, we're going to get a longer chain of amino acids. Right? So through removing water, we're going to be able to link our amino acids. Through hydrolysis, we're going to be able to break apart our monomers and release individual amino acids. And what is linking our amino acids after we do dehydration synthesis is the peptide bond. And so this is kind of what we reviewed, or at least talked about, on the first slide. Now, pay attention here, because the peptide bond is formed between the carbon of one amino acid and the nitrogen of an adjacent or second amino acid. Right? And we're dealing with the carbon of our carboxyl group and the nitrogen of an amino group. Right? So if you remember, we've got here's one amino acid with its R group. All right. Here is, and this is in a diagram on the next screen as well. I don't know what I just did. It wasn't good. Here is nitrogen, carbon, carbon, OH, R, H. Right. Here's the other amino group, and so what ends up happening here is this. Right. Here's the carbon, here's the nitrogen. Right. Here's the OH. We produce that H2O. And now what ends up happening is there is a bond that is then formed between the carbon and the nitrogen. There's a bond that's formed there. Right? And that bond is actually strengthened with this oxygen. We'll talk about this more in the next slide. But that bond is the peptide bond. Right? And that's what we've been talking about. And so we remove that water and we create this new bond between carbon and nitrogen that is strengthened by the oxygen. So here's what it looks like. Here's what it looks like. And I think I've got another view of this on this same slide. So again, here's the amino acid. Let me use the laser pointer because I can. Here's one amino acid. Here's another amino acid. Here's the carboxyl group of one amino acid. Here's the amino group of the adjacent amino acid. Here's the OH, here's the H, pop out the water, and here you create that peptide bond. Up until now, in carbohydrates and in nucleic acids, this bond has been formed through oxygen. Oxygen has been left behind to create this new bond. In a peptide bond, it's the carbon and the nitrogen that are directly forming the bond. The oxygen is used to actually stabilize the bond. Right? We're going to talk about that here in a couple seconds. But if we reverse this, just to kind of show this, right, if we reverse this, right, here's the peptide bond. Right? If we reverse it by adding the water back in, now we split this up. And so look at it from this perspective. Here's the here's the carboxyl group, here's the amino group. Right? In this version, we're using 2 nitrogens from here, and this is another variation off of a carboxyl group where the hydrogen's already missing. But it doesn't matter. All right, this is an amino group. This is still ammonium. All right, even though it's an H3. Remember I said that was a possibility. This is another variation of the carboxyl group. You still have H2O. You're still releasing that water. All right, and now you're still creating the bond between the carbon and the nitrogen. But look at what's happening with the oxygen. Because the oxygen also has a charge, right, and it's looking to still stabilize with things, it actually forms an additional support beam or support bond with the nitrogen. Right? It's forming a bond with that nitrogen. Think of it like a suspension bridge. Right? You've got point A and point B, and the bridge is linking those 2 points. Point A is carbon, point B is nitrogen. The peptide bond is forming the link between those 2 points. But a bridge by itself without support is going to collapse. And so what do? They put those big beams going up the side, and then they have the cables coming down that attaches to the bridge to provide support and integrity as the bridge shuffles and sways in the wind, because there's a lot of movement in those bad boys. They're not rigid. Right? They're not static. Right? Those support cables allow for that shifting to go on. It stabilizes the bridge as it's moving. The oxygen is doing the same thing. It's releasing these invisible cables, if you would. These electromagnetic cables that's binding and adhering to the nitrogen to stabilize the peptide bond, which makes these bonds harder to break. You don't want your proteins breaking down really easy. You want them to be really stable. The peptide bond does that because the oxygen is not used to make the bond. The oxygen is now used to reinforce the bond. Let that sink in. Hopefully that makes sense. I hope. If not, you can review it and always ask questions. All right. Okay, so we're actually almost done here. I only have 2 more slides to kind of look at with you. I want to talk about what proteins do before we actually get into more of the action. Let's have a conversation about what they do. So first thing you need to understand is that proteins are shapeshifters. All right, we've heard of shapeshifters in horror movies. We know that very often shapeshifters can assume the shape and the body and the persona of somebody else. You hear about these things in horror movies all the time. These are the biological shapeshifters. They can change shape. They can go from a primary structure to a secondary structure to a tertiary structure to a 4th degree structure known as a quaternary structure. And their shape determines their personality. It determines what they can or cannot do. We're going to look at that in the next video. Right? So first thing you need to understand is that these guys change shape. And the shape that they take on determines and defines what it is that they can do. Right? So what do proteins function within the body as? Well, they are enzymes. We're going to talk a lot about enzymes in the next video. Right? Proteins can engage in catabolic and anabolic reactions, which is what enzymes do. They help drive the metabolism or the overall biochemical process of the body, again, to maintain homeostasis. Right? We know that proteins are actively involved in muscle movement, right? Oxygen transport, hemoglobin, structure to the cells, to the tissues, and to bone. Right? So there is a cytoskeleton that is made up very often of a very thin filament called actin, as well as some other proteins. It's what allows your cell membrane to maintain shape and structure. Right? Aids in communication and signaling. So there are proteins on the surface of every cell surface. Right? What we call major histocompatibility proteins. Right? MHC1s that identify the cell and allows it to communicate with other cells. There are glycoproteins, there are glycolipids that are also proteins that function as things like chemical singulars. Right? They can signal. My God, I can't talk. They can signal other cells. Right? Not only do we find these guys as cell communicators on the membranes, things like antigens that can sit there and talk to other cells, but proteins also aid in our immune system. Right? MHC2 proteins, major histocompatibility 2 proteins, are what we find very often on things like lymphocytes, T cells, B cells. Right? Antibodies are proteins. It's what our B cells use to fight off viruses and bacteria and allergens. So if we think about this, enzymes function in every single system. Every single system automatically relies on proteins. But our muscular system does. Right? What we call actin and myosin, right, rely on proteins. Your muscles would not be able to contract without proteins. Our cardiovascular system relies on proteins in the form of hemoglobin to transport oxygen. Every single cell and tissue and all of our bones require proteins to be able to provide that structure and integrity to maintain shape, which supports its ability to do the function that it's designed to do. Right? Every single cell across all 12 systems rely on proteins for cellular communication and to sit there and say, hey, I belong here. Don't attack me. When those proteins fail to function properly, we call that an autoimmune disease, where your immune system does not, your T cells and B cells and your neutrophils, don't recognize the antigens that say, hey, I belong here. Don't come after me. Your immune system says, screw that. I don't know who you are, and attacks. That's an autoimmune disease. Causes inflammation, causes joint deterioration, causes swelling, causes pain and stiffness, causes deterioration of ligaments and tendons and muscle and bone. Failure to maintain these proteins have drastic impact. Your lymphatic and immune system is rooted in these. Every single system relies on proteins. You cannot escape it. You can try. It won't work. What are some shapes? What are some shapes? Well, this shape right here is our common enzyme shape, all right, or what we call a binding protein. All right, so things can bind to this, all right, and they're going to come right in here. This is where they're going to bind. This is what we call the binding site or the active site. We're going to talk about more about this in the next video. Right? But proteins can also be more like this, more like it's a donut shape, right? Where they extend through an object, and now they've got a tunnel, so things can actually pass through here. All right. So this is another shape. Right? We find this with in our cell membrane. We'll talk about this in chapter 3 when we look at integral proteins that insert themselves through the cell membrane to allow things to pass through the membrane. And there's a reason why we need that. This right here, these are fibrous proteins. Right? Long linear proteins, things like actin, right, that drives muscle contraction. Things like myosin that drives muscle contraction. Right, just to give a couple examples of them. Right, collagen is another example of a fibrous protein. We're going to talk a lot about collagen when we get into tissues. This is a globular protein. Right, this is a globular protein. Things like hemoglobin, right, where we put together 3, 4, 5, 6 smaller proteins into this globulous form. And so we come to the end of part 1 of our look at proteins. You got an idea of what a protein is now and the diversity within those proteins. In the next video, we're going to look more specifically at their function. Right, we're going to look more specifically at their function. But I do want to round up. I do want to spend some time. Let's fill in our table. All right. So we're going to come back in here. This is like your cool down. I'm going to bring it back down. Let's do gray. All right, so again, we know we have carbohydrates that we've talked about. We know that we have nucleic acids. And now we can add proteins to our list. Right? What's the monomer and the bond and the functional group for carbohydrates? Can you fill it in? Think about it. You got 10 seconds. Go. If you answered, which I hope you did, a monosaccharide, you are correct for the monomer for carbohydrates. If you answered glycosidic bonds, you are correct. And if you mentioned that the functional groups involved in that glycosidic bond are 2 hydroxyl groups, you are also correct. What about nucleic acids? Fill in the blanks. 10 seconds. Go. And so if you answered the monomer of a nucleic acid is a nucleotide, congratulations. If you mentioned that the type of bond that is formed is a phosphodiester linkage, you are once again on fire. You are correct. And if you mentioned that this bond is created from a phosphate group binding to a 5' carbon and a 3' carbon on surrounding sugars, you are also correct. What about proteins? What's the monomer, the bond, and the functional groups involved in these guys? 10 seconds. Go. All right, did you answer the amino acid as the monomer of proteins? Did you mention peptide bonds as being the bond type? And did you mention that peptide bond was formed between a carboxyl group and an amino group? If you did, you are swinging through the ring of fire and not being burned. Good job. We're going to build on this, keep reviewing it. If you missed anything, go back and review that. We're almost there, guys. Almost there. And hey, until we come back again, I'm going to catch you on the flip side.